Yep. So my talk will have a full component today, and okay, let me see whether I can. Yes. Okay. So the first part, I'll share my understanding of Alzheimer's disease and the cholinergic transmission. The second part will be showing my background research on nicotinic receptor function in the hippocampus. The third part will be my Reg Scholar pilot project and what I'm trying to do in this pilot project. And the last part will be my future direction and other ongoing collaboration happening in my lab. And so since everyone here are experts in Alzheimer's disease, I'm going to go through the first couple of slides really quickly because Alzheimer's disease has been first identified by Dr. Alzheimer about 123 years ago, and the first patient is Augusta Dieter, and her prominent symptom is memory loss, and later followed by a language deficit, mood swing, and disorientation. And pathologically, you saw pronounced atrophy of her brain, and under the microscope, you observed optical dense plaque, and inside around the neurons, and inside the neurons neurofibrillary tangles. So after more than 100-year research, we have identified those optical dense plaque composed by the aggregate of amyloid beta protein, protein beta 42. And the genetic study has shown if you overproduce A beta, then you will lead to accumulation of A beta plaque in the in human brain and in the transgenic animal models. So definitely amyloid is, I think, the star hypothesis for the Alzheimer's disease. And for me, as trained as synaptic physiologist, I'm looking at the synapse. What does A beta do? So amyloid protein, A beta actually released from the presynaptic terminals. And they're shown to enhance glutamate release from presynaptic to enhance the synaptic transmission. However, when you had a high level of amyloid proteins, you sort of lead to the dysfunction of synaptic transmissions. So currently, most therapeutic efforts are centered at a reduction of amyloid proteins in the brain. And sometimes for some like BAC inhibitors, the patients receive the trial drug as show the loss of cognitive functions. And another thing I noticed from the report is those new amyloid reduction medication, they're slowing down. Is there a question from someone? Okay, I'll go ahead. So slowing down the cognition decline, but not stop it and reverse it or enhance it. So and another puzzle is people with amyloid protein not necessarily will have cognitive function decline. They could have normal cognitive functions. So because of those evidence, I like to introduce you to another hypothesis. So of course, the front center will be A beta and the tau, phospho-tau. So I want to introduce you guys to the cholinergic hypothesis. Why does this have any connection to the Alzheimer's disease? So actually, cholinergic system was connected to the human memory by pure, I wouldn't say pure accident study. So psychiatrists interest was testing the cholinergic receptor blocker, scopolamine, and when they administered to young healthy adults, and they showed the memory loss and a cognitive deficit just similar to the Alzheimer patients, which leads to the starting of the cholinergic hypothesis. So later, when pathologists examined the Alzheimer patient brain, they found a selective loss of cholinergic neurons in Alzheimer patients, as highlighted in this yellow bar, especially in entorhinal cortex and the temporal lobe. You see, for the normal healthy aged adults, you have like in, let's say, in the temporal lobe, you will have like this 43 artificial unit. And for Alzheimer patient, you will reduce to only 4.7. That's, you sort of lost 90% of your cholinergic input into hippocampus. And similar study was, another study looking at a nicotinic receptor, they found the reduction in Alzheimer patient as well. So definitely there's a reduction in the cholinergic transmission in the Alzheimer patients. And so people hypothesize, if we elevate cholinergic acetylcholine level in the synaptic terminal by blocking acetylcholine esterase, would that rescue or restore the cognitive function? And yes, they did. And this would be the like older generation Alzheimer drugs. They're aiming at blocking acetylcholine esterase and to elevate acetylcholine level at terminals and to sort of rescue cognitive function. But this had nothing to do with either plaque or phospho-tau. And how can this rescue cognitive function, which is the first symptom for the Alzheimer patients? So this requires scientists to do more research to find out how acetylcholine connect to the cognitive functions. So before I get into that, I want to talk a little bit about cholinergic synapse. So as depicted in this diagram, so at the presynaptic terminal, they synthesize acetylcholine by transfer acetyl-CoA to choline. Then it's released upon stimulation. And they will cause the synaptic cleft and activate the cholinergic receptor. So there's two kinds of cholinergic receptors. One is muscarinic receptor. They're a G protein coupled receptor. You can tell by the size. That's not my research interest. I'm interested in nicotinic acetylcholine receptors. They're pentameric ion channel, and they allow sodium to come into the cells and depolarize the membrane. And there's a two main subtypes in the hippocampal neurons. One is called alpha-7 homomeric receptor. The other is alpha-4 beta-2 heteromeric nicotinic receptors. So they differ in their properties. So you can see alpha-7 not only allow sodium to pass, they also allow calcium to pass the channel, enter the cell. And as we know, calcium is sort of a main secondary cellular molecule in many types of cells. And besides that, alpha-7 receptor has a really fast desensitization. The black bar indicate the presence of acetylcholine, but the receptor quickly desensitized. However, for alpha-4 beta-2 receptor, they will stay open. They do not desensitize that fast. And also, they had a different sensitivity to the blocker, and alpha-7 receptor will be blocked by alpha-bungal toxin and the MLA, and the alpha-4 beta-2 will be blocked by the DH-beta-E, which I will mention in the later of my talk. as a tool to select the subtype of nicotinic receptors. So at left hand, it's a table summarized by Dr. Levine. Actually, he's a professor at the Duke Psych department, and he has summarized nicotine's effect on the memory learning behavior task across different species. Those are normal fish and rat and mice. So they all show enhancement upon the administration of a nicotine. And additional, a clinical trial, I think, conducted more than a decade ago, has shown that if you gave a memory impaired patient with a nicotine patch for 6 months, and they also showed improved the paragraph recall and the delayed word recall. So those evidence showed that a nicotine can enhance learning and memory, but how it does. And so this will lead to second part of my talk about my research background on nicotinic receptor functions. So I started research, this research project to looking at how nicotine regulates glutamatergic synapse transmission of multi-fiber pathway. So we choose hippocampus because that's critical region for learning and memory. And at the tree synapse circuit, dentate gyrus will be the first relay neuron to allow information to pass through and start the learning process. So the question is, the first set of questions I want to ask, how does nicotine regulate in which receptor is mediated this effect, since there's two subtypes, and what a site of modulation is at a pre-sub terminal or post-sub? And the third would be, what kind of intracellular signaling is activated? Is calcium alone or calcium activated protein kinase? So to answer this question, I used acute slice, hippocampus slice preparation, as shown here. You will see the glass pipette patch onto the CA3 parameter neuron. Then I recorded the current by electric stimulation of the dentate granule cells. And you will saw this downward current represent the response of evoked by the glutamate release. So if we base the slides with the nicotine with alpha-7 modulator NI1738, you saw the enhancement of the peak amplitude of those currents. And actually those currents remain enhanced for quite some time. It's better shown in the histogram here. So later, I used a selective antagonist for alpha-7 to confirm this effect. It's mediated by alpha-7 nicotinic receptors. And also this effect is absent in the alpha-7 knockout mice. So as we know that alpha-7 led calcium into the neuron, then calcium can activate calcium-dependent protein kinase. So we use pharmacological approach to block PKA, where KT5720, or CAM kinase 2 with KN62. We saw inhibitor effect of a PKA inhibitor, but not for the CAM kinase 2, which indicates this enhancement is mediated by the protein kinase A. And so what electrophysiology data has shown us is alpha-7 activation will lead to PKA. So we hypothesize that it's this PKA activation will depend on calcium, and which will activate calcium, activate adenosine 1 and produce cyclo-MP. So to prove our hypothesis, we took a newly fluorescent biosensor allow us to direct image those two signaling molecules. So we use a GCAM to image calcium, and use a FRET sensor for cyclo-MP. So another advantage for imaging study will tell us the location as well. So this is a cultured hippocampus slice preparation. We infect the dentate granule cell with a GCAM where it emit a green fluorescent, and we did a counter infection in the CA3 with M-cherry. Then if we zoom in the emulsifier forming terminal onto the pyramidal cell dendrite, you will observe those green giant emulsifier terminal, which can be as big as 5 micron, allow us to do imaging, unlike other type of pre-sample terminal, as indicated by the arrow. So when applied, I think sorry about that, the wording for the agonist alpha-7 directly selective agonist will show the increase of the calcium signal, and this was absent in the alpha-7 knockout mice. And this provides evidence that alpha-7 receptor is present at the pre-sample terminal and allow the calcium to enter into the pre-sample terminal to enhance the glutamate release. And secondly, we used the cyclo-MP sensor to detect the production of cyclo-MP. So now we use this alpha-7 select agonist choline, which also is many ingredient in those vitamin and supplement as well. It's the same, and choline is actually selective alpha-7 agonist. Then we saw the so when the alpha-7 bind to cyclo-MP, you will reduce the FRET, so as shown here. So this downward reduce of a FRET ratio represent the rise of a cyclo-MP. And this cyclo-MP production was blocked by the alpha-7 selective blockers. And we also identified adenosine-1 as the enzyme produced through the siRNA study. So I hope I convince you that activation of alpha-7 enhances glutamate release. And this is dependent on the production of cyclo-MP and activation of PKA. I have later also demonstrated that will lead to the phosphorylation of synapsin and increase release probability. So next question we ask, also it's sort of asked by the reviewer of a paper, it's like, do you think there's a cell type different dependent reaction, the signaling of alpha-7 activation? So there's a mixed population of neurons in hippocampus, in dentate. Let's say there's the main type will be the dentate granule cell. There are glutamatergic neuron, they release glutamate and depolarizing the next cell and elicit firing. So their counterpart will be the GABAergic interneuron. They release GABA and hypolarizing the next neuron and inhibit the neuron firing. So because having those two types of neuron, you will keep the excitation and inhibition balance that allow you to convey the message reliable. So we started this project thinking they should be identical. There's why they're different in any way. So we did repeat a similar experiment in those two neuronal type. So we measure the cyclo-AMP rise in the dentate granule cell, and we saw the same rise, and which everything is good, we're happy. However, when we did a measurement in the GABAergic neurons, we saw no changes in the Fred ratio, which indicate no production of a cyclo-AMP. And what's wrong? And so we go back to our old method, electrophysiology, and we patch onto the dentate granule cell and the GABAergic neuron, and we saw the alpha-7 current. As reported many times, the alpha-7 receptor it's highly produced in the GABAergic neuron, which, as shown, they had a peak amplitude can be 1,000 picoamp, and it's much bigger than the granule cell. And so definitely receptors are there. Why there's no production of cyclo-AMP? So next thing, we check the calcium transient activated by the alpha-7 receptors with GCAM. And first we examine the granule cell. As you can see, the flash of the calcium signaling showing here. And everything it's as we expect. And you have both in the soma and the processing, they had a huge, robust calcium transit. So we performed similar experiment in the GABAergic neurons, but they had for the soma they had some change, but it's to much lesser extent compared to the granule cells. And so why they had a more receptor, they had a bigger current, why they had a less calcium? And we hypothesized because they had so many calcium buffer protein expressed in the GABAergic cell. And if we raise the external calcium concentration from 2 millimolar to 10 millimolar, we should see the calcium transient to overcome the calcium buffer capacity of the GABAergic neuron. Indeed, when we increase the external calcium level to 10 millimolar, we saw the robust calcium response. Also, we saw the cyclo-AMP production in the GABAergic neuron. They do have ability to produce cyclo-AMP, but under the normal calcium, external calcium concentration, they do not produce that. So that would generate a different effect in vivo. So we, by collaborating with Dr. Go-Hong Cui, we built a in vivo fiber photometry system, as shown here. And so we inject AAV virus, express GCAM and the TD2 metal in the hippocampus, then also implant the optical probe, then we record a fluorescent signal through spectrometer. And as you see this in the open field cage, the mice, this white is tethered to this white cable. And then we record the spectrum data emitted from the GCAM, and then calculate the calcium signal from different cell type. And so what we found that if we administrate nicotine where IP, we observed an increase of neuronal firing from the dentate granule cells. As shown here, the recording, this is 1 minute, each recording shown here is 15 minutes. And this enhancement is absent in the alpha-7 knockout mice and indicated it's mediated by the alpha-7 receptor. And for GABAergic neuron, and we did not see this effect. Nicotine actually doesn't change in the firing rate of GABAergic neurons. So they had a peripheral effect on the glutamatergic neurons. So to summarize this part of talk, I hope the evidence sort of support that alpha-7 receptor mainly on working on the glutamate receptors, and they were enhance calcium signaling at the prereceptor terminal and produce cyclin MP and activate protein kinase A and enhance glutamate release. And they had a much lesser effect in the GABAergic neurons. So, and this will lead to me to my current pilot project as REC scholar. So the question I'm asking, that's under physiological situation. That's how nicotine works, and nicotine receptor produce their function. But what happened to the Alzheimer animal model or Alzheimer patients? What happened? What kind of change? What changed? So for animal model, I choose ApoE animal model instead of traditional overproduction of a beta model. So the reason is they're more related to the physiology, because I think there are more than 7 million Americans. They're carrying the 2 copy of the ApoE4, so it's more relevant compared to the familiar Alzheimer, because that's only affect 5% of the Alzheimer patient populations. Another reason I choose ApoE4 model, because they had a early synaptic pathology where really mimic Alzheimer patient as a synaptic physiology. That's where I want to study, so that's why I choose it. And there had a reduction of LTP in hippocampus. And another note says they had a selective loss of GABAergic inhibitor neuron, and showing the early neuronal hyperexcitability. And so the last and the most important reason I'm interested, there's some link showing the nicotine receptor deficit in ApoE4 carry, although it's indirect link evidence. So in this bar graph, they did some learning and memory tests on the ApoE3 double copier and ApoE4 the double copy carrier, and they applied a nicotine wear nasal spray, and they found the nicotine did not have much effect on the ApoE4 carry, but significantly enhanced sorting accuracy and this PM accuracy. Like they had a pre-warning showing this kind of card going to show up, and both accuracy. So the hypothesis, the ApoE4 patient carry, they might have a deficit in the nicotine, reduce the nicotine receptor function. So that's why when you give a nicotine, you enhance this dramatically, so they show this kind of behavior. Similarly, in transgenic mice, they show that ApoE4 had a reduced LTP compared to E3, as shown here. And however, this can be mimicked when the wild-type mice was subject to the nicotine receptor blocker, MEC. So they're producing the reduction in their LTP, its nicotine receptor dependent LTP portion is lost. And also sort of indirect evidence showing there's a nicotine receptor deficit in the ApoE4 genotype. So, however, we don't really know what happened for the nicotine receptor by the ApoE genotype. And this is another study I cite, but unfortunately it been retracted last year. And they show that in the Alzheimer patient, you will have a enhanced A beta and alpha 7 interaction. And they're arguing that ApoE4 sort of indirectly increase. This one it's got a retraction, so we can pay less. So based on those evidence, we hypothesized that ApoE4 reduce the function of nicotine receptor in GABAergic neurons in the hippocampus, which because this nicotine deficit will lead to neuronal hyperactivity and impaired synaptoplasticity and A beta accumulations. So I plan to test this hypothesis through 3 aims. So first aim, we like to just determine the level of the alpha 7 receptor expression in vivo. And we're going to build up the ApoE3 and the E4 mouse colony and collect their hippocampus from 6 months and 12 months. And we're going to perform cell type specific quantification with RNA-scope. And also we're going to stain of a cholinergic marker chat. And then we're going to try to correlate those findings with age, sex, and AD pathology such as neuronal loss and amyloid plaque. I want to share with you one of our polymer data with RNA-scope staining. So before I go into detail, I want to tell you the key for the green signaling is alpha 7 nicotine receptor messenger A. The red would be the GAT2 messenger A labeled GABAergic neuron, and the magenta will label all the glutamatergic neuron. And the Dalby staining shows as blue will label every cells. So you can see in the panel A, there are many, the magenta signaling cover the entire slide, means there's mainly a glutamatergic neurons in the hippocampus. You will see the red neuron scattering. That's GABAergic neuron that keep them in place, do not overfiring and become too decided. And as we zoom in the dentate region, we'll see some of the this yellow arrow showing one of the glutamatergic neuron express highly of alpha 7 messenger RNA. However, they're not located in the molecular layer where granule cell. So this is we suspect this could be a MOSI cell, and we're getting the messenger RNA marker for the MOSI cell to identify the identity of this glutamatergic neuron. And also you'll see among the GABAergic neurons, they have a differential level expression of alpha 7 as well. And look at this cell, they had a really high level of alpha 7 expression, and this neuron will have much less. And we're also trying to get a GABAergic subtype marker like SST neuron, parabolic GABAergic neuron marker to separate them in the future. So for our second aim, we want to look at a functional effect. And so to do that, we're going to use hippocampal culture, and we're using AA virus with a kinase 2 promoter will allow us to label the glutamatergic neuron, and we also use a CRE-ALF system to label the GABAergic neuron. Then we're going to look at the spontaneous firing of glutamatergic neuron and GABAergic neuron as shown here. The blue represent glutamatergic neuron, and the GABAergic neuron always firing at a much higher frequency, which has been documented for so many years. Then we're going to look at the nicotine agonist effect, antagonist effect to probing the nicotine receptor function indirectly. And also we plan to do electrophysiology to directly record nicotinic current between those two types of neurons. And for our last aim, we like to evaluate change of nicotine receptor in the brain sample of APOE4 Alzheimer patients. And currently I have no preliminary data for it, and really need help to get this going. And this is also a request from the reviewer of my grant proposal. They said in order to, they want to establish the relevancy to the Alzheimer, to the human, because sometimes there's a huge difference between animal model and the Alzheimer patients. So for that, I plan to maybe get a few brain sample and to perform the RNA scope study to look at the changes between maybe different sex, age, and AD stage. So I think I still have 5 minutes, so I'm going to use that to tell you what else going on in my lab. So one of the platform I'm trying to develop is neuronal function screening for the Alzheimer risk factor. As I read, there's many theory about Alzheimer because for even double copy of an APOE4 carrier, not everyone will develop Alzheimer, right? And there's a double hit theory, so they maybe had another risk factor will lead to the development of Alzheimer. For that, I plan to use culture from cultured hippocampal neuron from either normal syphilitic plaque and APOE4 and E4, E3 mice. Then we expose them to the different risk factor has been found in patients, and then to look at what be most impactful for them, and through like neural development and neural activity, and also nicotine receptor function. And then we can pick out those risk factor combinational and to do detailed electrophysiology at a circuit level and maybe in vivo recording. And also we can test out the novel therapies to mitigate those risk factors. And one of the risk factors is this collaboration with the investigator at RTI, and they're interested in cyanotoxin, which produced by the cyanobacteria during the algae bloom. And as we're facing the climate change, this become more and more relevant. And so we're trying to use the equipment InQ site at RTI to start this building the platform. And we're using the concentration where the CDC deems the cutoff 10 nanomolar, really low, and they deem it safe level. So when we put on the neuron for 30 days, as indicated by UU1, which labeled the dead cells, you'll see exposure for the two type of cyanotoxin, which has MCLR and a nodulin. You'll see there's a huge increase in the green signaling and the count. So, and we're going to continue this project that I felt very interesting. What would happen if we test on the APOE4 neurons? And since they had like I think lost ability, that had a lesser capability for neuronal repair and handling the stress. And so this could be one of the compounding factor for the APOE4 carrier. And also another topic, toxin we test is ethanol, and which we get interesting result. And when we look at total cell death, we saw the significant neuronal cell death, cell loss. However, if we look at neuronal, we did post life death assay with conjugated with neuronal marker staining, allow us to do cell type specific the cell death assay. So we found actually there's no change in the neuronal numbers. So it felt like the increase of the cell loss are mainly due to the loss of a glial cell, but not neuronal cells. So we're going to repeat this as well. And so this, before I end my talk, I like to thank my lab member who did most of the work, Trolada. She did those the high school screening with InQSite. Yahoo did most animal ApoE4, E3 animal work, and Kristen, she did the work immunostaining for those in vivo recording photometry related work. And of course, I like to thank my mentor and collaborator, Dr. Yanko, who provide all the virus, AAV virus I used in my project. And as mentioned, for this cytotoxin project, it's collaboration with Dr. Leslie Cologne at RTI. So, and this is the funding source. And thank you for your attention. I will open to the questions.
Awesome. Thank you so much. Can't get over how nice the how clean your RNAscope images look.
That's a good question. We're just started this. I think the signal actually pretty good.
Yeah.
However, we haven't. Oh, I'm in the process getting a software to quantify those dots. You know, we don't. At the moment, I don't really have the software to quantify each green dots, and in the cell body, that's another thing. Another thing is those reagent from RNAscope. It's I think it's outrageously expensive. It's only like for 20 sections, it's like $3,000. So we're trying to also test a reagent from other company, which I heard it's much cheaper, but I don't know the quality. But I'm actually pretty happy about the staining of RNAscope so far.
And you know, do you think that those measures are, I mean, sort of semi-quantitative? Yeah.
Yeah, I think so. So some of the published paper, they're saying they're the measure the dot and the size of the dot. Mm-hmm. Then you can also use how to say the area accumulate. No, it accumulate like it's for this. If you select ROI, you can say, hey, the green signal cover how much area. Mm-hmm. That's another way to compare. But if you had a very high level messenger RNA, it might look like a GAT2. They're all complete carried, so you have to use intensity. So, but that's not the case for alpha-7. So, and so we're working on quantification, and I think we're trying out the different software at the moment.
And then thinking about the potential aim in the REC project to look at this in some human specimens, are there existing maybe like single cell sequencing data sets that could be leveraged to sort of get an idea on this, or is the sort of spatial aspect really going to be kind of critical? Like if you're able to map cell identity back to the dentate and, you know, to, you know, the various cholinergic or GABAergic neurons or whatever it is that you wanted, do you think you could potentially look at the alpha 7 expression in those to try and draw some conclusions, or would you really need like, the sort of spatial transcriptomics approach, like what I think the ADRC funded some pilot work with Simon Gregory toward that, although I don't know that he was looking necessarily at the brain structures that you were primarily interested in.
I think a single cell seed would be helpful and powerful. It just, the reason is I don't have the expertise in that area, so that's was not proposed. But it would be powerful, and also it's we don't have the ability to do at the central, so that's another reason I didn't propose. But I think it would be powerful. Those are new technique, and it would be complemented to the structure as well, I think, and certainly worth looking into. That's what I saw. So
yeah, that's fantastic. I see we have a question from Heather. I'll go ahead and read it because it's in the chat. So Heather asked, are nicotinic acetylcholine receptors found only in the cell membrane, or are they also present in the intracellular membranes in the neuron, such as the mitochondria?
I'm not aware the nicotinic acetylcholine in the mitochondria, but they're due in the intracellular organelle, and their expression and sending to the outer membrane, it's tightly controlled by two chaperone protein, natural, and another one. It just slip out. So it's highly controlled by the chaperone protein as well. And another thing I like to point out, alpha 7 is expressed everywhere, not only on neuron, but also in the astrocyte, glial cells, and they have been shown to regulate inflammation response. But I'm the synaptic person, so I didn't present. I was not expert in that aspect. So
yeah, I just think it would be interesting. It could still be important at the synapse, but because some of the genetic information is suggesting that some of the genes involved in protein trafficking are so important in the disease that it would be interesting if that's where these particular kind of receptors come into play.
Yes, those chaperone, two chaperone, Rick and the natural, is certainly on my radar and the list. So because why the ApoE4 can reduce nicotinic receptor expression, it could be like they provide less lipid. There's no lipid around, and for them to hang on it in the membrane. So there are multiple possibility, but I just want to see for sure. Is there less? The unfortunate thing for the nicotinic receptor is we don't have a good antibody. So the current like there's a paper back 2 years ago showing test a 10 antibody, they had a similar staining in alpha 7 knockout and the wild type. So that's why most people were avoid antibody. We're using the functional assay to look at the receptor and the messenger RNA. So that's a backstory, but I certainly think that chaperone protein could play a huge role. That's a good point.
Thanks. Interesting work. Thank you.
Thank you.
Any other questions? I had one more if I'm not taking anyone's spot. Okay. Maybe you sort of touched on this, but I don't know. Are there different sort of transcript isoforms of CHRNA7 that might be important here, or potentially expressed and processed differently in different neuronal cell types?
So are you asking there's a splice variant for alpha 7?
Well, different isoforms, yeah. If there are different, yeah, I guess either protein isoforms or at the RNA level, just different splice variants that might have different expression patterns within diverse sort of neural cell types.
I'm not really aware of that. I think that another thing link alpha-7 to mental disabilities. There's a deletion in, I think, chromosome 14 where alpha-7 gene located, then you had a mental disability. That's another evidence, but I'm not aware of isoform wise, other mutation. I can look into that. That's something I can look into that and see so relevance. I think that's a good point. That's a good point.
I'm just curious, obviously there's different acetylcholine receptors, and in work that I did years ago, we're looking at nicotine addiction, right? And so we have genetic risk loci, like from GWAS, that are in CHRNA5, CHRNA3, but we saw a lot of heterogeneity across different racial and ethnic groups, and we always wondered or hypothesized if that was related to alternative splicing and how that might work.
So I didn't work on the addition. I think one of my lab mates did. I think addition is different subtype other than where I think it's alpha-3, a nicotinic receptor expressed in different regions. I think it is habenula. It's a different part of brain where controlled addition, because we're nicotinic lab at IEHS, so people are looking at addiction and also the weight regulation by nicotine, because when you smoke, you're slim, when you stop, then you gain weight. So people are looking at a different aspect of a nicotine receptor. So I think alpha-3 is another subtype people have been studied mainly with addiction and weight loss. So, but in the hippocampus for the memory, I'm interested mainly alpha-7 and alpha-4 beta-2. So.
Well, we're about out of time. I do see Marianne put a note in the chat, just highlighting the importance of the work to understand the mechanism of action for pesticides and different organotoxins in the brain. I do think that it's both relevant and grant writing wise, reasonable to be thinking about, savvy, I guess is the word, to be thinking about how some of these risk factors might relate to climate change and other things like that. There's a big effort, at least at Duke, going on in the sort of climate change effects on health outside of the sort of standard infectious diseases space. And so yeah, I'd certainly encourage you to continue that line of inquiry.
That is a great question, because most of pesticides, they work on the insect nicotine receptors, and they had some potency on the human nicotine receptor for sure. And this could be one of the risk factor compounding, let's say ApoE4 carrier. And I think this is a very good suggestion. We can try out on our platform and see what happens. And like you said, maybe we can aiming for the agency who interested in those kind of work. So this is a very good suggestion. Thank you. Thank you.
All right. Well, we are at 2 minutes past, so I think we'll go ahead and wrap it up. But just wanted to thank our speakers so much for giving a really great talk and walking us through a fascinating story that you've built over the last several years. And we're really excited to have you as one of our new Rec Scholars and watch you push this work forward and stand ready to help you out however we can.
I'm looking for
please reach out.
for that. Thank you. Thank you for everyone, the good suggestion, and hope we build a successful project. Thank you.
Fantastic. All right.
Okay.
Thanks, everyone.
Thanks.